few words about the work that they do and the collaboration that we're involved in tonight, and hopefully more in the future.
Can we speak?
Okay, thank you. So, hi folks. So my name is Michael Walsh. I had the privilege of serving as chairman of the Biomedical Engineering Division for the last year, and I kind of just relinquished the role about 2 hours ago. So I'm kind of cheating a little bit here. So just Engineers Ireland, it's a professional body for engineers in Ireland. There's like over 24,000 members, and it's kind of unusual setup. It's made up of 11 regions, which are basically geographical parts, like there's a Cork region or West region. There's 9 societies, so they're kind of like a heritage society or young engineer society. And then there's 11 divisions, and they're kind of broken up by disciplines. So those would be like civil engineering, and one of those is biomedical engineering division. Now within that then, the biomedical engineering division has 4 kind of parts to it. There's the academic, industrial, the rehabilitation, and the clinical based engineers. So it's one of the kind of newer parts of engineering. It's the fastest growing area of engineering. And the reason for that is there's so many terrific problems that we need to solve. And part of what we're trying to do as engineers is to figure out not only solutions to the problems that we have to face, but also the methodologies to try and figure out how to solve these problems. And that's part of what Paddy's talk is going to be about tonight, is the use of numerical models and trying to solve all these problems. So I'm delighted that this is the first in the line of talks in conjunction with the Alchemist Cafe. We hope to make this an annual event. So I'm going to hand back to our host, and she's going to introduce our speaker today, who's probably, if there was a Mount Rushmore of biomedical engineers in Ireland, he would face to be one of those chiseled on it. So I'll hand back. All right?
Great introduction there. Okay, so thanks, Michael. So as I said, and as Michael said there, this is the first time that we've collaborated together, but we really would love to collaborate more in the future, as it's exactly the kind of organisation that we'd love to get involved in, and to let the public know more about what's happening in research and how it actually applies to their daily lives. So just to introduce an idea about what the Alchemist Cafe is, can we have a show of hands of anyone who's never been to an Alchemist Cafe before at all? Brilliant, loads of people. Okay, so just to give you an idea of the general idea of what an Alchemist Cafe is, it's part of the Café Scientifique movement, which was started originally by a guy called Duncan Dallas in the UK in Leeds in 1998. So since then, there's over 30 of these kind of events being held in the UK on a regular basis. And the idea is that you get somebody who is an expert, or perhaps has written an interesting book on a particular subject in an area of technology, research, engineering, maths, computing, and get them to come in and have a chat with general public audience over pints, over coffees, and just that the public can get to interact with this person, but also interact with each other about what they think about some of the technologies that are being developed, and maybe some of the implications for society of these technologies, some hot issues like things like stem cell research, or any of those things where the public have a particular interest in them. So overall, in terms of the people we get in, we get them from the arena of biology, chemistry, maths, and engineering, as we have here tonight. So we try and cover all areas of science, and to keep the cafes as informal as possible as well. So that tonight, the cafe, we're going to have roughly about 20-minute introduction from our speaker, and then we'll we'll have some complimentary finger food and refill our glasses, have a chat about what we've just heard, and then we'll get together again and discuss what we were talking about. Maybe, hopefully, our speaker will be able to take some questions as well at that stage if anyone wants to ask him anything. Okay, so just to introduce our speaker tonight. Our speaker is Professor Patrick Prendergast from Trinity College Dublin, or I hope you won't mind me calling you Paddy.
No, Paddy,
yeah, okay, so just to give you some information on Paddy. So Paddy originally got a BA in mechanical engineering, and a PhD in mechanical engineering from Trinity College Dublin. He was elected as a Fellow of Trinity College in 1998 and a member of the Royal Irish Academy in 2008. At the moment, he is Vice Provost and the Chief Academic Officer of Trinity College Dublin, and holds a chair in engineering. And he still remains an active researcher himself and is a principal investigator, currently holding a Science Foundation Ireland award titled Computational Mechanobiology, Fundamental Advances and Implementation of Medical Device Engineering, So when you hear that medical device engineering, you know there's going to be some really interesting and intriguing applications of what Paddy's going to tell us about tonight. So without further ado, I'd like to introduce Paddy to bring us into the exciting world of mechanobiology and engineering.
Well. I've never given a talk like this in a pub before. I think I've given maybe 150 talks in various seminars and conferences around the world, but never in a pub. I sang in a pub maybe, and never with a pint of Guinness in my hand, so this is going to be unusual for me. Mount Rushmore, my God, that's something. I don't think I should be on Mount Rushmore, but I think Professor De Puer, who's the real leader in the beginning of Irish biomedical engineering, should certainly be. It's great to see you here. Alchemist and the Alchemist's Cafe. I think I'm going to start with Alchemist because, as you know, one of the most unusual people that I found was an alchemist was Newton. Newton was an alchemist. I did all kinds of things trying to make diamonds and trying to make gold and didn't succeed, at least not in getting physical gold anyway. Newton was an alchemist. And Newton was also the person who came up with really forces of the kind we're going to talk about here. Because this mechanobiology that I'm going to talk about refers to how forces have their effect in the biological world. And when I was thinking of how I might start this introduction, I thought of Newton and his famous quote that he said, "I do not know what I may appear to the world, but to myself I am only a little boy, paddling on the seashore, with the vast ocean of knowledge out there before me." And that was Newton's famous quote. And I was at a reading by Derek Mahan recently where he had something to say about Newton, and a poem called Research. So this is not what you expected, all right? But I'm going to read you this poem. It's called Research. "An actual conch, like a human head on its side, washed up and left here by the ebb tide. A magical sculpture, perfectly arbitrary, lies as if dropped from orbit, or they will launch research to find ice in the sea of rains, a first dubious twitch of mud and plants, signs of life on the other planets, whispers of inchoate mind and flickering brains. Meanwhile, on Earth, we've mud, plants, pleasure, pain, and even real lives to be getting on with, seasons for this and that, the works and days of many mice and men, as Hesiod says. Best to ignore the great ocean of truth, the undiscovered seas of outer space, and research this real unconscious conch on the shore with its polished archaic face and its air of myth." And I guess what Derek Mahan is saying in that poem is, if you just reach down and pick up one of the shiny pebbles on the seashore, you've got enough to be getting on with in your research. And even looking at one of the shells, you don't have to wade out into the vast oceans. Just touch the thing nearest yourself, and there's enough to be going on with in your research. And that is what I'm going to talk about, the research when you reach out and touch your neighbour there, the research that we do on the human body itself. The human body, we often say, us who do biomechanics like me, is a machine. A machine created under the action of forces. Forces are everywhere, we always say. Forces are, when Kieran is leaning against the bench, there's a force. But have you ever seen one? You see, the thing about forces is you don't really see them. They're like virtual in a way. You can see their consequences, you can see their effects, but you don't really see them themselves. And of course, we only see their effects, and it's like that a thing, you know, there's this, I've come over here from Trinity, from the front square, where I work at the moment, and there's a famous Trinity philosopher, Berkeley, or Berkeley. It's very interesting in Trinity whether you say Berkeley or Berkeley. It defines what side of Trinity you're on. Anyway, I say Berkeley usually, except when I'm talking to people who might like to hear me say Berkeley. But he said that a thing could not be seen unless didn't exist unless it was perceived, unless a human being perceived it. But then how, of course, did some things exist if there wasn't a human being look at it? If there was a tree in front square, but there was no human being looking at it, then did it not exist? Of course, the trick out of this is that, well, God is looking at it, and God can see it, therefore it exists. So this is the problem we often, it leads me in a very roundabout way to the issue that I want to talk a little bit about, is that engineers are often very reluctant to do experiments in the virtual world, in virtual space. Yet forces, you've never seen a force and you're happy to believe that they exist because you see their consequences. And when we create simulation environments for carrying out experiments, it's a bit like that. We're doing an experiment in this virtual world, and we have to be able to trust the outcome of these experiments in some way. Some people are more trusting than others, but by and large we have to realize that there's a lot of things we don't see that we believe exist. And the same with conducting experiments, so-called numerical experiments. Now, forces exist and affect the human body in a couple of ways. Like when I walk around like this, my muscles generate a force, and I'll have if I could do breakdancing here, I probably would. And you can see the consequences of forces on the musculoskeletal human frame. In fact, it was a Neapolitan mathematician called Borelli who first put forward the idea that the human body was a machine in this way. He was a student of Galileo's, and he wrote a book called De Motu Animalium, on the movement of animals. And I had one of my most scholarly thrills, actually, when I wanted to see this book in the original, Latin. And I went to Trinity's library and they didn't have it, but they told me that it could possibly be in Marsh's library. I don't know if anyone's ever gone up to Marsh's library. And I went into Marsh's library and I said to the librarian there, a lovely woman, have you got the book by Borelli, De Motu Animalium? I don't suppose you have, but I'm here on the off chance. And she said, we have 3 copies of it. And she knew exactly where to get it. And she took it down from the shelf. And I wanted to open a particular page, and she had to cut the page with a knife. And that book had been in there since the 1700s. And I was the first person to open it. And I looked at the diagrams. I got a great thrill from that. But this was radical for the time, because Borelli was proposing that the human body was a machine. But it's what he didn't say, because it wasn't current in the science of the time, is that actually it's a surprisingly good machine. Not only can it move around and do all the things we do in it, generate its own force and energy and so forth, but it's actually a self-repairing machine. And when you put mechanical forces on it, it cracks and tears will generate in the tissues, and the machine can repair itself over time. And this repair capacity for organic forms, most organic forms, the human frame included, is regulated by mechanical forces. So this is a subtle consequence of mechanical forces. They regulate tissue processes ongoing inside in the body. And this is what we talk about, what we mean by mechanobiology. Mechanobiology, of course, is a made-up word. Doesn't even exist in Wikipedia or any of these dictionaries, I've noticed. I'm thinking of starting a little wiki page on mechanobiology. I noticed that many of my colleagues in Trinity have wiki pages named after themselves. I think that's very, very interesting as well. Couldn't resist that, but I won't do that. But if anyone wants to start one, they can. But mechanobiology then is looking at how mechanical forces regulate the behavior of tissues, regulate them, maintain them, and actually are involved in repairing them as well. So how does this happen? How does this self-repairing machine respond to the action of mechanical forces? Well, it happens through cells. Cells are the sort of action elements distributed throughout the tissues. And there can be thousands and millions of cells, even in a square millimeter of some tissues. And these cells respond to mechanical forces, some more than others. And what we do in our research, and some people are here that are involved in this research, maybe Dave will be able to say more about what we do in the question and answer session. But what we try to do is simulate the activities of these cells within tissues. So we're going to get a little bit technical here now. We create a lattice, and each point in the lattice can have a cell or it can be empty. If it has a cell, then it can have a cell of various different phenotypes. And that cell can engage in cell activities. It can move around the lattice, that is, it can migrate. It can proliferate, that is, it can divide and multiply up in the lattice. It can change its phenotype from one type of cell to another within the lattice. It can die within the lattice. So we have to imagine a domain with this orthogonal lattice, some points occupied by cells, and eventually indeed all points occupied by cells, and you have these cells moving around inside in the lattice. Now the stimuli that the cells receive within the lattice depends on where they are in it. They will receive different mechanical stimulation within the lattice. And we can simulate thereby how cells, as they move around inside in this lattice, how they differentiate, how they synthesize matrix and generate different tissue types. Now the difficult part of this is designing or discovering, or we sometimes say, the algorithms governing how the tissues respond to the mechanical stimulation. Because proliferation of the cells is going to be regulated by mechanical stimulation. The change in phenotype is going to be regulated by mechanical stimulation. We have to make some hypotheses about how these cells respond to mechanical stimulation, code that up into an algorithm, combine the algorithms for the various different cell activities into a simulation, and run it and compare it with experimental outcomes. How long have I talked for now? Okay. So this is where we create a virtual environment for conducting experiments, because we can create a domain, fill it up with a lattice and seed some cells into it, and then apply mechanical stimulation. And that mechanical stimulation often comes in through mechanical devices. This is where the medical devices come in. So I've got a few devices that I'm going to pass around here that I borrowed from Bruce Murphy there about half an hour ago. And these are stents. You can open up that one and pass it around, and here are more. You can put them onto your hand if you want, and you can see different kinds of devices. Pass that around there. Here's another one. They're cardiovascular stents. When you implant these stents are implanted into tissues, into an artery, to prise it open and hold it open. Usually, if you're in danger of getting a heart attack, often it's because downstream of a blockage, there's not enough oxygen coming to the tissue. So you have to prise open the artery and hold it open with a scaffold called a stent, and that will allow the blood flow. The problem with this is that you're applying very unphysiological mechanical forces to the arterial tissue when you put in this stent. And the tissue doesn't like that. The cells within the tissue are unphysiologically stimulated, and they respond to this. And actually sometimes they can respond, and the response to the stent can be worse than the initial problem, and you can reform the blockage around the stent. This is the sort of thing we want to simulate. And we don't want to simulate it as a scientist might for some disinterested curiosity reason. We want to simulate it because we want to have a virtual environment that can tell a good stent from a bad stent. We want to be able to indeed reduce the number of animal experiments carried out to test stents. Animal experiments are no good for medical devices usually. If there are people here doing drug research, I know that you use animal experiments a lot, and there's nothing wrong with animal experiments in the right place. But they're not often good for medical devices because the structure of the organ is different in the animal than it is in the human. Oftentimes, indeed, if you want to do an experiment on a test a thing out, another reason to do numerical experiments actually on devices for humans is it's very difficult to do controlled tests with humans. You can't blind the surgeon or the interventional cardiologist into which kind of stent he or she has implanted into a patient. They'll know, and therefore that confounds the result interpretation when they're asked to do it. Oftentimes you can't blind the patient either to the kind of implant they've had. All these reasons make it more important to do computational simulations on how medical devices perform. I had 3 hip implants here, which I wanted to pass around as well. We're going to have a little competition. We'll ask some non-engineers. Are there any non-engineers? Yeah, Belinda's a non-engineer. To just tell us which one of those 3 do you think is the best hip implant? I've broken the rules by stepping over the line here. But I can pass them back to some of your other Alchemist Cafe colleagues. The point is, it's very difficult for engineers in assessing how implants behave to do it in animal experiments or in bench tests. If we could get robust, believable virtual worlds, virtual environments for testing implants, it would be extremely beneficial to the medical device industry, and that's why I guess Science Foundation Ireland have funded our work, our principal investigator grant on this subject. So that's the reason for trying to do these computer simulations. Also, we want to do them for tissue engineering reasons. I think you've had probably previous talks on tissue engineering where you want to simulate how an implant behaves, a biomaterial scaffold seeded with cells behaves when it's implanted into a patient. We also want to do computer simulations of that to try and optimize the seeding density for cells and the structure of the scaffold to maximize the speed of tissue formation in the implant. So I'm coming very close to the end. I just want to say a little bit to finish on virtual environments and numerical experiments for computer simulation. I think these have great potential to be used in medical device engineering. But the problem with them is that clinicians don't really believe them. And in fact, biologists in general have a very skeptical view on computer simulations. And I wonder why this is. And I wonder if in a way we couldn't have a discussion about this. Why do biologists have such a poor opinion of computer simulations or of virtual environments? Engineers generally have a good view of this, but I found myself working in bioengineering. You're trying to interface between engineering and engineers who believe in models and theoretical models of systems and biologists who are extremely skeptical of them. And I'd be interested to have a discussion on how this gap can be better bridged. So, have you got your answer?
We have a vote for which one most people in the audience who are non-engineers thought was the best one. They thought this one. Probably not right.
Yeah.
We wouldn't vote for this one.
You wouldn't vote for that one? Huh?
No, one vote.
Oh, one vote.
Yeah. So, and then this poor fella didn't get anything.
This is a Muller prosthesis. And you're right, it's the worst. It's called the banana by those in the business because of the shape of the stem of it. It didn't do very well. It's got too many sharp corners. This is the one invented in the 1980s. It's called a Charnley prosthesis, and actually it does very well. Much better than many that came on the market afterwards. This is an interesting one because it's different for the left and the right. It's a luminous. It's made out of titanium, and the marketing ploy really to make one different for the left and the right. It doesn't do so well as this one does. So you actually got the right answer even though you know nothing about engineering. No need for computer simulations.
Just votes.
Just votes. Democracy works. So the cardiovascular stents that we passed around, and we've quite a few people in the audience here that are working on stents, and their opinion as to the value of computer simulations in design of cardiovascular stents will be very interesting. I'll just finish by saying it is quite important, stents, you know. 85% of the cardiovascular stents in the European Union are manufactured in Ireland. So we have a very big cardiovascular stent industry. So it's appropriate that we're leading the research on computer modelling of how cardiovascular stents behave when implanted into the body, carried out there by Colin Boyle. So there you go. I'm finished now, Belinda.
Yeah.
So happy to
Thanks a million.
go to the next phase.